Look, good evening, everybody. We'll just wait for a few to join us. We're just at the 7:30 mark, and then we'll kick off. Couple just trickling in now. Just give it another minute to see if we get a few more attendees, and then we'll kick off. Everyone's obviously running back from the tractor tonight to get to get here for 7:30. Brill. Well, I'll get going just so we're not eating into everyone's evenings. So good evening, everyone. Thanks for joining tonight for the NSA and Moredun joint webinar series of reducing the risk of reproductive diseases in sheep. My name is Nicola Noble. I'm the project manager here at NSA and a sheep farmer in the Cotswolds. We've got a really interesting lineup of speakers tonight to discuss some of the most prevalent reproductive diseases that are available in sheep. We've got Alan Murphy from the Animal and Plant Health Agency. We have David Longbottom from Moredun. We've got Lee Innes from Moredun, and we've got Beth Wells from Moredun and Sarah Thompson from Moredun as well, helping with the Q&A, etc., and doing things behind the scenes, putting lots of resources in the chat. So if you could keep the chat for sort of general chit chat, as I say, Beth and Sarah will be sharing some really useful resources in there on the topics discussed today. And for any Q&A sort of burning questions, can you put them in the Q&A box at the bottom, just so we don't miss them? As I'd hate to sort of one get put in the chat and then there's quite an interesting discussion going on and it's sort of missed. If you could please put your questions in the Q&A, that'd be great. So our first speaker, Alan Murphy, from the Animal Plant and Health Agency. Alan qualified in 1990 and spent 16 years in the private practice of North West England, concentrating on livestock species. He was awarded a diploma of bovine reproduction during this time. He left the practice in 2006 to join the Veterinary Laboratories Agency, the VLA, based at Sutton Bonington. He studied for a further qualification with AFA support and was awarded an MSc in Applied Toxicology from the Surrey University. With the pending closure of Sutton Bonington, he returned to a private work as a consultant for a few years before rejoining AFA in 2018, and is now based at the Thirsk Veterinary Investigation Centre. So Alan, I'll hand over to you.
Thanks, Nicola. Right, I'm the warm-up man. So from that perspective, you get me for about 10 minutes, which is good because my accent's not always that easy to follow. So from the perspective of what am I going to talk about tonight, it's really just to give you a bit of an intro as to what Lee and David are really going to focus on from the problems of what's actually going on. The idea is we're trying to sort of look at abortions and reproductive loss. Now, as you can see from this, ovine abortion, they come in a variety of different bags. We don't have any particular loyalty to any particular feed company. We get them in all sorts of shapes and sizes, and they do come in all sorts of shapes and sizes, quite literally. Mineral buckets are also another popular option. Now, I've been working with sheep for 30 plus years. To say at least there's a degree of pragmatism, pessimism, dark humour always has to be applied to sheep. The fact I was always told at an early day they spend most of their life looking at the ground, working when they can get into it at the most inappropriate time, is not an untrue fact that has to be said. But loss is loss, and we're not in this business to basically lose them. We're in this business to hopefully get them over the line to where we want them to be, to breed from them, to finish them as fat lambs, etc., etc. So we do want to know when something goes wrong. But it's important for us to actually find out what the actual problem is. And the problems can be found out in a variety of different places. APHA has gone through a lot of structure change over the years, and if we go north of the border to Laswade, Moredun's there, and St. Boswell's up there as well. SRUC do as much work as we do in terms of sheep abortion investigation, along with all other species as well. But that diagram just shows to you there's lots of options for you to get help. Let's support investigations, etc. And that's also supported by private practitioners who can use us, they can use private labs, they can use all sorts. But the basic idea is if you think you've got a problem, pick up the phone and talk about it and at least get it investigated if it does trigger some alarm bells. Because it's important to know, not only for the fact of what potential actions you might need to take going forward, but in addition to that, what actions you can take. They're not always lost causes. And it is important to get that kind of stuff to us. Now, we're obviously looking at reproductive loss tonight. Now, reproductive loss in its own right is a big one. You actually look at the potential issues. We say to you, okay, well, if you've got this kind of figure, that kind of figure, it becomes very difficult for people to make a suitable sort of challenge, like say, okay, well, if I've maybe got 2 abortions, I'll get them looked at. I've had one farm and they get every abortion looked at. I've had another one only trigger off at the 10th abortion. Everyone's got different trigger points. However, you should maybe try and be a bit more pragmatic about it in terms of saying, well, what is a trigger point that's useful? In HDB, they do a fantastic amount of work and research going on to this, and it's a good comprehensive approach. It's not specific to a farm. It's looking at the overall industry in terms of saying, well, these are the figures you should be looking at in terms of what you're looking at for your MTUs, lamb losses, and these are all things that should flag concerns and issues saying that have actually got issues with regards to a variety of diseases, not just abortion, not just maybe stillbirths, when we're looking at lamb losses, etc. But looking at the abortion, which is what we're looking at tonight, this is reproductive loss, remember. We're focusing on things at about 2%. Now, 2% for a flock of maybe 100, that's not very many in terms of the actual numbers. It could be 2 ewes that have been bumped and knocked, and actually they've got 2 abortions. Suddenly that becomes 2%. But if you've got 1,200 sheep running around, it takes quite a lot to get to that 2%. But by that stage, you could be right in the middle of it. So timing can sometimes be moved a little bit as to how proactive you want to be, how good your control systems are in place already, and when do you think you actually want to look. And the reason you're looking is because you're in a business. You want to basically make that business profitable as best you can. There's a hell of a lot of factors out there already pushing down the margins in terms of how we operate as things stand now. We certainly don't want to add to by introducing or potentially trying to cope with a disease that we could have done something about. And if you look at the costs, they're all out there. Now, these costs are empirical. They're not specific to your farm. It's pretty much a figure that's made up as a sort of, this is an accumulative number, and we've averaged it out and done some numbers of this. But if you consider that 30% of the lamb losses we have are due to abortion and stillbirths, irrespective of how good the market is or bad the market is on Monday, you want to be at the market with live lambs. And if you haven't, because you're in that 30% grouping, you've got something you need to do something about. The cost of abortion, that varies. Did you do it by AI? Did you just do it with sort of like broken mouth yow that's pushed out a couple of lambs and thinking, no big deal? There's still not a zero in front of that pound sign until you get after the decimal point. It's a cost, and it's a cost that you could have probably done something to prevent if you've got potential diseases that have got diagnosis made, and therefore you can do some control. Now, we're going to be talking about enzootic abortion tonight, along with toxoplasma. The graph I'm going to show you at the end skews the figures a little bit, but we all know the reason why that is. But the basic premise is if you're looking at over 10, 20, 30 million pounds a year due to enzootic, and believe me, it does happen. It's a variable figure depending on how much the incidence is, but it's a phenomenal amount of money to lose. And it's quite simple to actually control. And it's very well marketed and documented how to control it as well. But yet we still do a lot of things in how we manage our sheep. You think, that really doesn't make sense. Why are we doing that? So we need to get better at how we control these diseases. And controlling them is quite straightforward, because if you actually look at how we introduce them, there's a lot of them. And these are just the infectious ones I'm looking at. I'm not even covering other stuff. We'll mention that in a little bit. But you look at the bought-in sheep, it's phenomenal as a marketplace. And people buying things here, there, left, right, and centre. Markets do this, markets do that. Oh, I just got a few sheep off my friend because it was easy. You've no idea what the concept is. I was around in 2001. Biosecurity became a byword for everybody. We seem to have maybe misplaced it a little bit in the past few years. We don't seem to worry too much about it. We really should. It's important that we consider what things we're bringing in. And these are diseases that aren't just a disease. If you consider, okay, well, I'm going to maybe control enzootic, or I'm worried about Campylobacter, what else has it got? Has it got scab? Has it got foot rot? The list is endless. So we're moving on from reproductive diseases to other things. Toxoplasma in cats, that's a whole different world in its own right, because it's not just that cat that's pooed for the past 2 weeks and it's going to give you toxo. There's other issues with toxo that are vastly more complicated that people need to be aware of. Birds, are we feeding them on the ground? Are we feeding them in troughs? Are we feeding them inside? Are we feeding them inside? Yeah, they're good. Well, actually, are they good? Because there's other issues you can get with vermin, etc., that can spread other diseases just as readily. Then how do we feed them? Are we feeding them silage that's been made as a big bale? Yeah, but it was a big bale. It looked really good when we put it in the ring feeder, but then the sheep have been standing on it for about a week. Listeria is everywhere in the soil. You're going to paddle it in. Other things go off as well, mold and things. We seem to think that ruminants, because they've got a forestomach, are pretty much indestructible. They really are not. And if you're pregnant, you really are not indestructible. You're vulnerable. More importantly, the fetuses within you are vulnerable as well. So you need to look after them as best we can. So good quality feed, good quality biosecurity, good environments, good health status overall. Keep them healthy. They'll look after you. And more importantly, they'll look after the lambs as well. But how do we look at it when we've actually got the problem? Well, the diagnostics are straightforward, and this is not a complicated thing to do. Getting this material in to have a look at is pretty much the first challenge. It's okay, so I've got too much on today, I can't get them in. You can't really run the risk of not getting this stuff looked at. Now, the problem you might say is, yeah, but you're civil servants, you only work Monday, Tuesday, Thursday, Friday, you reason up on Friday, you do bugger all. That's not really the case, but there's private vets out there as well, willing to get this stuff done, get some samples, get some material to them, they can get it off to private labs and get it to us. You can at least get diagnostics made. Samples are straightforward, don't need much. Placenta's kind of important. Now, we know that quite often you think, oh, placenta's been trampled into the straw, I've not got a hope of finding that. Fair enough, that's maybe a lost cause at that point, but it is a useful sample to get hold of, and if we can, we can. Fetuses, we do like a fetus, it's pretty useful, we can get a lot out of it, but not if the collie's eating most of it, or not if a badge has been found out in a fell side and you bring it in, it's a lamb that should weigh about 2 kilos and it's about half a kilo because its insides have been sucked out. That's not really much use. So be pragmatic of what you're going to bring to us to have a look. But the vets, most of them can find what a spleen, the liver looks like and differentiate the two of them. There's a fairly good idea where a brain can be found. These are all quite straightforward things to get. Blood sample in a ewe seems a bit straightforward, but it can tell you an awful lot if you've got an abortion problem going on, especially with Enzootic toxo. And even the ewes, if ewes are dying due to that abortion issue or they're aborting at the time, it might be a fact you're looking at sick ewes, and the reason why they're actually pushing lambs out is because they're sick in themselves, which is a major thing that people seem to forget. It's actually a sickness in the ewe that actually quite often will then get the lamb to abort. The infection has to go through the ewe in the first place to get to the fetus. So therefore, if the ewe's been off colour, and all you've got is a fairly sort of rotten lamb, it might not be much benefit bringing me the rotten lamb, but if the ewe's died, that's probably quite useful for people to have a look at. So consider the bigger picture in terms of what the actual problem is you're dealing with. And when I say it has to be looked at, these samples can be done anywhere, to be quite honest with you. You don't have to be too precious about how you're doing them. You've all been raised in the world of CSI and silent witness, and everybody thinks you're going to be looking at all these things in some fancy, complicated environment. Yeah, fair enough. My office looks a bit tidier than most PM rooms because it's a built structure for the purpose of it. Excuse me, it's got tables, it's got running water, it's got a roof, which is a great thing to have, to be quite honest with you, if you're trying to do PMs out in the farmyard. But you don't have to be too pristine about these things because at the end of the day, it's a sampling exercise to see what you can find and see what's relevant to your infectious process. And therefore, you can use fairly lower council tax band pit kit as well. Doesn't have to be fancy, doesn't have to be smooth, but it needs to be something you can actually get a look at and get a decent working progress into in terms of getting the vets to get the samples and then do it themselves. And farmers will do it themselves as well in terms of just taking the samples. There's lots of guides out there. You even have to get the vet for an expensive on-farm call-out. Just take the samples, bring them in, or take the aborted lambs down to the vet and get them to do it. You need to investigate. We need to look at these things. At the end of the day, we know what we're aiming for. It's to try and control these things that we're seeing on a regular basis. The numbers on the side there goes up into the hundreds and 700s. That's the diagnostics that we make. The very, very small grey bands you see at the top of that graph are basically DNR, diagnosis not reached, because basically the sample wasn't good, it might not have been infectious cause, the sample quality was pretty problematic. But you can look on there and see that the fact that the two subject matters we're going to talk about tonight are toxo and enzootic. They're not going anywhere. They are regular offenders in terms of how we see our abortion protocols fitting in, and Campylobacter is a bloody close third. 23 to 24 season, however, gave us a lovely dose of Schmallenberg again, which seems to be coming in cycles every 5 to 6 years. Now that's skewed the books in terms of actual numbers and representation. Now that's the disease that where we can recognize and we see what the problems are. But what we should also do is not forget the fact that we shouldn't be putting dustbin diagnosis in. If you got an abortion and people say the lamb looked a bit slow or a bit dodgy or it came out, and it doesn't mean it's Schmallenberg. It could be something else. So therefore, maybe have a look at them. And remember, APHA and SREC do free testing. So at the end of the day, they're willing to look at to samples and see if they can exclude Schmallenberg. If they can exclude it and you've still got a lamb with them, they could then do some testing on it. At least you've jumped over the hurdle of getting the lamb to someone in the first place. So think about these things as we go forward, because toxo and enzootic, CAMP in particular as well, they're not going anywhere. And we need to get better at how we control them. And more importantly, we need to use the methods of control that are out there, which we're going to be looking at in a while. There's other problems in there as well. Whilst the infections are all on the left-hand side and fairly obvious, you need to remember that there's a lot of other problems that can actually affect the ewe. Fetus is there to be carried by mum. If mum is not 100%, she will expel the fetus to survive herself. As such, never forget the ewe's role in this place, in this situation. Other conditions don't have to be infectious. They can be problematic for the ewe herself. We recently had a case this year where the people had severe problem with scab and a bought flock. They basically bought the farm acquired a hefted flock, for whatever word, they couldn't get rid of the scab, so they decided to dip heavily pregnant sheep. Under whose advice, I have no idea, but they lost 10 ewes sets of lambs due to the fact that ewes don't like being dipped and they're heavily pregnant. But that was their sort of open rationale to try and control scab. So there's other problems that sometimes manifest themselves in terms of what will give you an abortion. So just bear that in mind. If you don't get a diagnosis, it doesn't mean that there wasn't the infection that was causing what the common things of enzootic toxa. Talk to the vet, talk to the diagnostic lab, make sure they're getting the big picture on the farm, because that's important as well. This is what we're aiming for. You want live lambs to go whichever direction they're going in, breeding, finishing, whatever, doesn't matter. But I want them running around and springing, not coming in a feed bag, because in a feed bag, I know my day is going to be not necessarily a bad one, but I know someone else has had a very bad one. So on that note, I shall leave you to the bit you've been waiting for, which is Lee and David, who are going to talk a bit more detail about the two primary ones we've looked at so far in terms of just touching on it. The big ones are Enzo and Toxo. So with that, I shall leave you to it and hand back to Nicola.
Great, thanks, Alan. It was really good food for thought and a great scene setter for our next two speakers. Before we move on, I've got a quick question. Is there an easy way to find out like the rough cost of sending a lamb for a post-mortem through APHA or a diagnostic through APHA?
Yeah, gov.uk test lists are all listed in terms of prices. The only thing I would say is that some practices have a handling fee or an interpretation fee, whereby they look at my results and try and jazz them up to make them more important. They don't. But at the end of the day, sometimes people have that aspect. So I can't account for what private practitioners might add to a handling fee in terms of reports, generating, etc. But our fees are all listed. It usually goes under two codes. Either for sheep, you're looking at what they call a TC11 or TC12. But if you go on the gov.uk website, look for APHA diagnostic price lists. It's all listed, and I'm sure you could probably get a hyperlink fitted into the actual Q&A systems. And at the end of the day, it's all listed in terms of open-minded. That's the cost. That's what APHA will charge the vet, what the vet might charge on top of that. We have to deal with vets. We can't deal direct with farmers. We have to have a contract system with the practitioner. That's just the way we operate. But that's how the system works.
That's wonderful. I know that Beth's already shared some links to what diagnostics can be done through APHA in the chat. So please have a look at that if you're interested. So our next speaker is David Longbottom. He's a scientist based at Moredun. He graduated from Manchester University in 1895 with a BSc Honours in Biochemistry from the University of Edinburgh, and then in 1989 with a PhD in Biochemistry of the cholera toxin. He's then worked as a postdoc at the MRC Human Genetics Unit at Edinburgh on cystic fibrosis and moved to Moredun in 1993 to develop vaccines for controlling nematode infections in sheep. In 1994, he moved to chlamydia research, initially developing better detection systems for chlamydial infections of farm livestock and zoonotic infections. He also has an interest in bovine respiratory diseases and specifically in the control of Pasteurella infections, as well as genetic investigation of microbial populations associated with specific disease syndromes and monitoring environmental health through sensitive wildlife species and the dynamics of livestock. Very, very diverse CV here, David, lots of different avenues to go through. He's currently the editor and a board member for 3 scientific journals, and he leads the EU Discontrols Expert Committee, on chlamydiosis, and is president of European Society of Animal Chlamydioses and Associated Zoonoses. So a very, very highly renowned professional scientist here, specifically in enzootic abortion. So I'll hand over to you, David, as the expert, to give us a bit of an update on Enzo.
I should actually add first, Nicola, that I've handed over the presidency now of the European Society. But anyway, that's fine. Thank you very much for that introduction. And thank you very much, Alan, as well, for your great introduction. I'm going to talk today, as Nicola has said, about enzootic abortion. I'll just share my screen if I can. It was all going so well. Sure. Okay, hopefully you can all see this.
Yeah, we can see that now, David. It just should go into presenter mode in a moment. There we go, perfect.
Okay, right. Okay, so I'm going to talk about enzootic abortion. It's also known as chlamydial abortion, ovine chlamydiosis, EAE, OEA, whatever you want to call it, but it's all enzootic abortion. This is typically what you would see on a farm with maybe one live lamb, a couple of dead lambs, and so glossy looking placentas. If you can recover those, that is useful, as Alan has indicated, for diagnostic purposes. This is the bacterium responsible for the disease. You can see in this picture, and it's changed its name over a course of years. It used to be called Chlamydia psittaci, then it became Chlamydophila abortus, and currently it's called Chlamydia abortus, and hopefully it'll stay as Chlamydia abortus. If you look at the right-hand picture, this is one single cell that is invaded by one single organism of chlamydia. It's this small one here called an elementary body. And that gets in, and then it multiplies and it fills the whole cell. The cell bursts, and the organism goes on to then infect neighboring cells. And that's what causes the infection and ultimately the disease that occurs in the placenta. So Alan gave you a chart of year by year of the most common diagnosed infectious causes of reproductive loss in sheep. And over the last 5 years, just need to move these away. That's it. Over the last 5 years, chlamydia is, generally speaking, being the #1 cause of infectious abortion, about 37%, apart from the 2023, where it actually, I think it was in 3rd position after Toxo and Campylobacter. But nonetheless, it's one of the 3 main infectious causes of abortion. Oh, sorry. Yes. So as well as sheep, it affects goats, but it can also affect other livestock, pigs and cattle, as well as deer and horses, and even other animals. So it's important to remember that the pathogen is zoonotic. It can infect humans. It's a rare event. I should actually stipulate it's a rare event, but it can cause fever, headache, nausea, malaise in infected people. It can cause spontaneous abortion in pregnant women. This is why pregnant women are not advised to go near lambing ewes. But unlike in sheep, where the ewe does not die, it cannot actually be life-threatening for the pregnant woman. And here I've got a picture of a farmer that died in the Netherlands. So this is at 26 weeks. And we can see here this coagulation of the blood in the uterus that resulted in death, unfortunately, that person. This little figure here is just to stipulate that chlamydia is one of a number of chlamydial pathogens that infect animals, including birds and cattle and horses, koalas. But it also, as I said, infects humans as well. So this is typically what the placenta looks like in an infected animal. The infection is silent. We call it silent. We cannot detect it at all in a non-pregnant animal. You can only detect it once the animal becomes pregnant. And the actually infection and disease generally manifests during roundabout day, well, it's after day 60, but it's usually detectable after day 80 to 90. But it's only detectable by killing the animal and checking by pathologists. So you wouldn't know that your animal is infected at that stage. Generally, as you all know, it results in late-term abortion, usually within the last 2 to 3 weeks of lambing. There's little or no warning. You may get a vaginal discharge, but you may get a vaginal discharge anyway. And that vaginal discharge could be full of chlamydia that's highly infectious. And as I said, this is the typical picture of the placenta. It's very necrotic. The tissue is very inflamed. It's swollen. It's edematous. It's thickened, full of water. And it sometimes has this sort of exudate on the surface, this sort of mucus-looking, yellow, straw-looking stuff stuck on the surface. And that is basically just full of chlamydial organisms. But this placenta, as Alan's mentioned, is actually highly important because it could be the basis for a presumptive diagnosis for chlamydia. This is unique to chlamydia toxo. Lee will talk about that shortly, and looks very, very different, for example. Okay, so what we know, lambs can be born. They can be born dead, they can be born weak. They can actually be born live and infected. And that is a concern with regards to transmission, because if you have a healthy lamb, you may not realize that there's an infection in your animals, and they could go on to infect other animals. We do know that an abortion in year one, it could only be 1 or 2 abortions, could significantly rise in year 2 with up to 30% of your ewes aborting. Following abortion, what we do know is the ewe itself is immune to further disease. They do not get the disease again, but they can become infected still, and they can still be infective, but they will not abort animals again. The ewes can continue to excrete organisms following parturition in a vulval discharge. This can go on for several days, up to 7 days, even past 7 days. Essentially, until the discharges have dried up, you should treat those discharges as being highly infectious. And this, as I said, is important. The shedding is important in terms of transmission to other ewes that are naive. So if you have an extended lambing season, for example, if you have an abortion early, it could be passed on to another ewe, and that ewe could then abort later on in the same lambing season, or it could abort in the following season, in the following year. So what are the sources of infection or transmission? Obviously, replacements are a big issue. Where you obtain those replacements from. Are they from a health certified flock? Are they from a vaccinated flock? You have to be wary of your neighbours, whether they're telling you that they've got Enzootic in their flock and whether it could be carried over to yours. We have no handle on the role of wildlife, whether it's birds, carrion, rodents, or any other vermin that might actually drag a placenta or carry it over into your farm and end up infecting, for example, your naive ewes. But in terms of an abortion event occurring on your farm, what are the infectious what is important in terms of infection and spread of infection? Well, it's the coats of the dead, the weak, and the live lambs. These could be covered in infectious organisms, and if a naive ewe comes and sniffs those animals, they can pick up the organism easily that way. I've already talked about the vaginal discharges, the placenta themselves, but also the bedding that the placentas or the animal or the vaginal discharges drop onto, all highly infectious. So infection can be transmitted animal to animal, as I say, from any of these things to a naive animal, so ewe to lamb, or even lamb to ewe. Estrus has been suggested to have a role potentially in terms of transmission, but there really is very, very little evidence of this. But what we do know is there's very, very little evidence of ram transmission, unless it's the mechanical act that occurs during sex, because chlamydia in sheep is not a sexually transmitted disease, unlike in humans. So how do you manage disease? Well, it's all about obviously preventing exposure within your flock. And what do you do if an abortion occurs? Well, you need to act quickly. You need to determine the cause and treat appropriately, obviously seek veterinary advice. If possible, collect the aborted material, the placenta particularly, into a clean bag, get a blood sample for sending to your diagnostic vet along with the placenta and along with the fetus, as Alan mentioned before. It'd be useful if you could isolate the ewe, certainly until the discharges have all dried up. Remove all infected material and carefully dispose of that by burning or disinfecting the area. You should also take care in handling all infective material. If you have disposable gloves, use those. Certainly practice good hygiene. And remember, these things are a danger for pregnant women and even immunocompromised individuals, and bear that in mind. In terms of disease control, it depends on the initial infection status of your flock. If you have a clean flock, it's about stopping it coming in. You want to keep your flock closed. You need clean replacement stock. I've already mentioned about purchasing from disease-free flocks. Knowing the infection status of your source flock. Even if they tested negative by blood, remember, these are potentially silent infections. You may not pick it up, particularly if they picked up infection during the previous lambing season. The antibody levels can be quite low in those animals. If they've aborted, the antibody level is generally quite high, and you can detect that. And it takes usually 1 to 2 years for that antibody level to drop off. We would always advocate vaccinating before mixing with your flock, particularly if it's unvaccinated flock. If it's vaccinated, we would still recommend that. But abortions can still occur in vaccinated animals. If an animal's already infected and you vaccinate, the vaccine may not overcome that infection in that particular lambing season. And that's something to remember. Even if abortions don't occur, you might still get shedding of some of the organisms in vaginal excretions on the coats of live lambs, etc. So there's still a risk there. If you have a small number of unvaccinated replacements that you're bringing onto your farm, there is a lower risk if the flock has been regularly vaccinated against EAE, but you should mark those and vaccinate them as soon as possible. And that is probably quite an important point currently at the moment. We'll be coming onto this later with regards to the shortage this year or the lack of vaccines. If you have an active infection, it's about preventing the spread, managing the infection, treating and diagnosis. And in terms of the diagnostics, again, if it's done by antibody-based, we have to remember it's hard to predict if an animal's infected. The diagnostic tests only confirm an infection. So if you have had an abortion, the diagnostic test will pick that up because the antibody levels are high. But if an animal has just become infected, you may get a small rise. And that's why sometimes we would advise taking 2 readings, 2 blood samplings for diagnostics, but closer to abortion where the antibody levels will be starting to rise. We would never advocate the prophylactic use of antibiotics. It's not appropriate. But under extreme circumstances, in order to limit losses, oxytetracycline can be used. But bear in mind, again, it does not prevent established infection. You may still get some abortions. You may still get organisms shed at lambing. Overall, though, vaccination is still the best long-term option, particularly if you've got existing infections. And we would always say seek veterinary advice on the most appropriate control measures for your circumstances on your farm. In terms of diagnostics, we've got a range of tests that we can use in our labs. Some of the veterinary laboratories use some of these approaches. I've mentioned looking at the placenta. Here we've got an infected one in the center here, where we can see a lot of infection going on in that particular placenta. And it gives us immediately an indication that this is EAE. You can take a small bit of that placenta, we can put it onto a microscope slide, and we can stain it with something called modified Zeal-Nielsen, and it shows up these little pink, you can maybe see these little pinkish dots around here, and these are chlamydial organisms. And that again gives us an indication. MSD stains a range of bacteria, but gives a good indication that it's chlamydia. In a pathology lab, you can actually do immunohistochemistry, and you can stain using specific antibodies to chlamydia. And here we can say these are what we call trophoblast cells on the surface of the placenta that are highly stained for chlamydia, all brown. That's full of chlamydia. We can also test by PCR, and a lot of the labs now do this. But serology is another option. As I said, it's confirmatory, not predictive. I've already mentioned after abortion, maternal antibody levels rise. This used to be the test that was used, complement fixation test. I'm not sure that this is used as much these days in labs, but it used to be the test that's used. The problem with it was it cross-reacts with another chlamydial pathogen called Chlamydia pecorum, which is even more common pathogen of ruminants and pigs than Chlamydia abortus. Excuse me. And particularly the enteric subtype, which is the most common infection in sheep. And this does complicate EAE diagnosis, and that's why we try to move away from the CFT. We developed a test that we commercialized back in 2015, which has much better sensitivity and is highly specific for Chlamydia abortus. But our test is still not predictive. It still wouldn't necessarily predict that you're going to have an abortion on your farm. It is generally still used as a confirmatory test post-abortion. I should also mention that none of these tests will, what I haven't mentioned yet is the vaccine side. I'll come on to that in a minute, but the blood tests do not differentiate between a natural infection and vaccinated animals. This is called DIVA compatibility. DIVA stands for differentiating infected from vaccinated animals. Okay. So currently, there are two types of commercial vaccine available, as I'm sure you all know. One of them is the live vaccine. It's based on a French strain called 1B. It's produced by SIVA and MSD, called CVAC chlamydia and Enzootic. And there's also a second type, which is an inactivated vaccine. And the only one that is currently available in the UK is one from Inmeva, which is produced by the Spanish company Hypera. This is a combined vaccine, so it's a combined inactivated vaccine of Chlamydia abortus and Salmonella abortus ovis. There are pros and cons in their use and limitations for all of these types of vaccines. Obviously, at the moment, number one would be availability, because there is none available at the moment. There's been a manufacturing batch specification issue by both companies. Again, I think we'll be coming on to this maybe afterwards. But essentially, we're hoping that SIVA will be able to produce a vaccine later on this year. MSD have said it'll be 2025. There's there's differences in terms of when you can administer these different types of vaccines. You cannot administer the live vaccines during pregnancy, for example, but you can the inactivated vaccines. There's differences in duration of immunity. So the live vaccines maybe give you longer immunity, maybe 2 to 3 years. The inactivated vaccines, you're advised to give a booster annually. Hence differences in terms of frequency. None of these vaccines elicit what we call sterile immunity. In other words, they don't completely stop shedding of organisms at lambing time. You can still get some infectious organisms coming out of the animals, as I said, on the coats of the live animals, as well as in vaginal excretions. And you have to remember and bear that in mind in terms of potential for transmission. And over the last 10 years or so, we've known now that there's a potential safety issue with the live vaccines. We have shown that the live vaccines can cause infection and disease in some animals. We still recommend to use the vaccines until any new ones come on the market. but it's useful to bear that in mind, particularly if you've got a closed flock or a flock free of infection. How do we tell this? Well, we can tell this because we've developed a molecular test. It's based on PCR and something called RFLP analysis. You don't need to understand that. But basically what we can do is we've got here wild type infections here. We've got the vaccine strain here. And these are 5 cases of abortion in the field that we've tested. And they've got the same pattern as the vaccine strain, which indicates that those abortions were due to the vaccine and not due to the wild type. So we can actually test now and differentiate between a natural infection and a vaccinated animal. So at Moredun, we're in the process of developing new vaccines, and we're obviously trying to make these as protective as possible, as affordable, easy to use. We don't want them to interfere with diagnosis, the so-called DIVA approach that I mentioned earlier. We want to try and reduce the shedding of these infectious organisms at lambing. However, I don't believe any vaccine will ever get rid of this completely. I have to be honest about that. And we want to make the vaccines as safe as possible. Again, we don't want them to potentially cause infection like the live vaccines. And that, again, is important from a zoonotic perspective as well, because of potential transmission to humans. So we're taking a number of approaches. We're taking 4 approaches at Moredun at the moment. I'm not going to go into any great detail of what these are about, but we're taking these 3 approaches at the top here, and we've got grants that are covering these vaccines. What I want to mention to you is this bottom one, C. abortus outer membrane preparation, COM-C. So what we have done, we've taken the pathogen, here it is. This is the infectious form of the bacterium, it's called an elementary body. And we can isolate this outer membrane here. I hope you can see my cursor moving around there, of the organism. And we can separate it from the contents. And that stops the organism from being able to infect anything we put it into. But it retains the natural structure of the outside of the organism. But that's great from an immunological perspective because it's mimicking the natural pathogen, and that elicits a better immune response then to the preparation that we've derived called the COM-C. So we have so far conducted 4 pregnant sheep trials over an 8-year period. We've looked at a variety of different things. We've looked at the amount of organism we put in. We've looked at the adjuvant that we add. The adjuvant is there to try and improve the immune response. So it's present in inactivated vaccines. It's not present with the live vaccines. You don't need it with the live ones. But ours is between a live and an inactivated vaccine. It's sort of in between. It's got the best of both worlds, if you like, but we have to add an adjuvant to it. And we can do a variety of different sampling and analyses in our lab to look at how effective the vaccine is in the field. So this is a summary of the 4 trials that we conducted. The only thing you need to be concerned about is this is the COM-C. If you look at part A, and look at the second column, that's the COM-C vaccine. The green indicates that we challenged animals after vaccinating them with chlamydia. So we purposefully infected them. And in the 4th column, you can see it's challenge control. We actually infected a group of naive animals, and you get about 50%, 30 to 50% abortion rate in those animals. What we're trying to do is improve that abortion rate with our vaccine. And you can see here with the COM-C, red indicates abortion, green indicates successful lambing. And we also look at shedding. The first column is one of the commercial vaccines. And we can see with the chlamydia COM-C vaccine here, that we've improved on the commercial vaccine and it's much better than challenge. So basically the COM-C vaccine seems to be quite effective at reducing abortions and reducing shedding. And we've conducted now three other trials to extend these studies, basically. And ultimately, we have found that we can give a single dose. The vaccine reduces abortion, pathology, and shedding. We've been in discussions now for a couple of years with our commercial partner, which is in Spain, called CZ Vaccines, and they're currently trying to commercialize the vaccine. And we're hoping it will be available from 2026 onwards. We're in the process at the moment of transferring the technology from our lab to theirs to scale it up to an industrial scale. And the vaccine company is applying, or has applied, for a limited market authorization to sell it within Europe with the European Medicines Agency, and also in the UK with the Veterinary Medicines Directorate. So that's just a quick summary of some of the work that we're doing. I can't stop without just indicating it's not just me. There's a lot of people been involved over the last 10 years or so that we've been doing this work. So that's just to highlight those individuals there. And I'll just stop sharing at that point. Okay.
Thanks, David. That was really informative. I learned a lot myself, and hopefully those listening have as well. And it's really good to hear that there's new vaccines being sort of developed and then trialed, and hopefully there is, positivity on the horizon for potentially another vaccine that's more, essentially better than what's already out there. So that's really good to hear. I had one quick question on you mentioned about labs that are doing diagnostics and where you can send placentas and things off for investigation. Is it similar to the situation what Alan was saying, where it has to be done through the vet? Did you have to? Yes, you have to send your samples through the vet to get them.
They're sent through the vet. Yeah.
Wonderful. So if anyone's interested, obviously, again, speak to your vet about potentially getting testing, etc. Brilliant. Wonderful. Thank you for that, David. Okay. So our next speaker tonight is Professor Lee Inns, also from Moredun. Lee's conducted research in the area of infectious disease of livestock at several different research institutes in the UK and Africa, with an interest in developing solutions to control diseases caused by protozoan parasites. She's an immunologist by training and has focused her research on developing and using vaccinations and methods to prevent disease. And more recently, looking at this One Health solution to improve health and welfare for animals and people. Lee's conducted research on Toxoplasma gondii for 30 years, working at Moredun Research Institute and collaborating with other researchers across the world. She's passionate about science, communication and public engagement and improving the health and welfare of livestock people through research and education. So I'll hand over to Lee. I think we're in great hands here to learn a bit more about toxoplasmosis.
Great. Thank you very much, Nicola. And thanks everyone for joining today. I'm going to try and share my screen. Let's see. Is that working all right?
That is, yes, Lee, we can see that.
See that? Okay, great. Great. So I'll just get that. Is that in slideshow mode, or are we?
It's still in presenter mode at the moment. So I think you just need to click on that.
Yeah. Is that better?
That's perfect. Great. Thanks, Lee.
Okay, well, thanks, everyone. What I'm going to do is talk to you about Toxoplasma, which you've heard from both Alan and David. It's probably one of the most important and frequently diagnosed cause of abortion. It's what we wanted to emphasize tonight is we're talking about the two main diseases that cause abortion. And they're very different in terms of transmission and how you control them. So toxo is one that sheep can become infected from eating Toxoplasma eggs. So Toxoplasma is a parasite, and I'll tell you a little bit more about how it's transmitted. It's quite an interesting one. But what the parasite does is it invades and infects the placenta, causing problems like abortion. But following infection, sheep will develop very good immunity against the parasite, and this does protect them going forward. And this is why the disease can be prevented through vaccination, which is probably the best way to control it. So I'm just going to talk to you a little bit about how it's transmitted so you understand how different it is from chlamydia that David was talking about. So the cat is absolutely central to the life cycle of the parasite. And usually it's young cats who go out hunting for the first time. They might eat a mouse or a bird that's already infected with the parasite. And in the gut of the cat, the parasite will develop, and then the cat will shed eggs. Now, I'm not sure if I get my cursor, if you can see that, but this is a picture of the egg stage here. And the other important thing is the egg is very, very resistant in the environment. So they can last in the environment for up to about 4 years. So they're quite tough. They've got a tough outer coat. They can also survive in water. So the route of infection for sheep is eating these eggs either in pasture or in feed. And they can also be transmitted to other animals, and we as people can get infected. It's quite an important pathogen for us as well. And we can also get infected from eating these egg stage, or if we eat tissues from animals such as sheep or pigs or cattle that are infected, you get the cyst stage in the meat, and that can infect us. So once the parasite gets into the sheep, it multiplies up. You can see these tiny little green banana shapes. That's the parasite multiplying in the cells of the sheep. And then the immune system starts working against the parasite, and it will insist. I don't know if you can see in there, there are hundreds and thousands of parasites here in these tiny cysts. And the cysts will stay in the tissues of the animal or in the brain, and they'll stay there for life. So that's just a quick run through the life cycle. So how does it cause abortion? So the main issue with your sheep is if they get infected for the first time when they're pregnant. So if they're infected when they're not pregnant, you basically see no clinical effect. Early to mid-pregnancy, you start seeing these mummified lambs or abortion occurring. If the infection occurs late in pregnancy, you might see stillborn or weak lambs that are infected with toxo. So I'll just show you a picture because, as David said, it's very different than from chlamydia. And this is a sort of classic situation you might see with infection at mid-pregnancy, where you see a mummified fetus, you can see there, and a stillborn lamb. But the placenta looks very different for the diagnostics. The cotyledons here, you'll see white spots. And this is areas of necrosis where the mother is trying to fight the infection in the placenta, and you get these white spots. This is sort of classic, you'll see for the diagnostics of toxo. So just to summarize, the infection is basically spread through Toxoplasma eggs spread by infected cats. Now the cats only shed for a couple of weeks, so it tends to be younger cats that are a problem, not your older cats. And once the cats have been infected and they spread the parasite, they're immune after that. The issue is that these eggs are very difficult to remove from the environment. So the other difference here is that Toxoplasma is not spread horizontally between sheep. So there's no risk of introducing Toxoplasma from replacement or bought-in sheep. That's another difference. They only get infected if they eat the eggs. Sorry, not eat the sheep. Eat the eggs off the pasture or in feed. You can get abortion storms occurring with this parasite if it's a naive flock that encounter the parasite for the first time. So like all of the diseases we're talking about tonight, early and accurate diagnosis is absolutely key. Dead lambs' placenta should be submitted to the labs, as Alan has described that really well. In the labs, they'll look for presence of Toxoplasma, and also they can take blood samples to look for antibodies. So if we're going to move on to what you're interested in really is how do we control this? So if we remember that Toxoplasma is transmitted through the consumption of these infective eggs. So it's really important to try and keep cats away from feed bins and hay. This is a typical situation of a cat in a farm. Just watch out in the feed sheds. You don't really want cats anywhere near there. We've got no treatments, unfortunately, that can cure sheep of toxo. We've done some research at Moredun showing that the decocchinate can be fed during pregnancy at a time when the ewes encounter Toxoplasma, which may reduce the disease. But the key research that's been done shows that once the sheep are infected with Toxoplasma, they will remain infected for life, but they develop very good immunity. And this has really helped in terms of developing a vaccine for Toxo. And there's a very good vaccine out there produced by MSD called Toxovax, and it's a live vaccine and it's attenuated, so it will infect sheep, but it will not persist in the sheep. So this vaccine has been tested thoroughly across the world. It's a live vaccine, so there are some issues with the short shelf life. It's really important that you use it according to the expiry dates, because if the vaccine is not live, it will not work properly. The storage conditions are also really important with this vaccine. I think currently there are no issues of availability of the vaccine, which is great, because I know there has been some issues in the last couple of years, but we're assured that there is no issues at the moment, which is really good. So it's recommended that you use this vaccine at least 3 weeks prior to mating, and this is because it's a live vaccine, but it's at least 3 weeks. You can use it before that. And we've also done research to show that it's a very long-lived immunity. So 1 dose will give protection for at least 18 months. So it's very effective. I'm just going to finish off with one slide here. We emphasize that this is an important zoonotic infection, so it will also infect people, and it's a major cause of disease in people globally. In fact, we think we've done some research to show that probably about 1 in 5 people are infected with this parasite globally, which is massive. So probably quite a few of you on the call may be infected with it. It's a major issue for pregnant women, and can cause real issues for if a woman gets infected for the first time during pregnancy, and for immunocompromised people. And the other interesting thing about the parasite is that when you become infected, it usually goes into your brain. Now, you may not show any signs of infection at all, but it will sit there quite happily in your brain. And there's some interesting research that people have done in Oxford looking at mice. Now, this is not a normal situation. This is more a normal situation with your cat and your mouse. But mice that are infected with Toxoplasma are not afraid of cats anymore. So you can see that the parasite's probably doing this to get into the cat and complete its life cycle, because you can guess what might be happening next. But some of the interesting research going on is whether, if Toxoplasma is in your brain, is it causing any behavioural changes? And I'll leave you with that one. And we've also got some information at the end here, which is we've got some fact sheets from Moredun and NSA, which you can get from our website. We've also got an animation, which is also available on our website if you want to get a bit more information about it. Okay, thanks, Nicola. I'll leave it there, and happy to take any questions. I'll stop sharing.
Brilliant, thanks, Lee. And that's fascinating, especially the sort of the level of engagement with human interaction and sort of the behavioural side of things. That's really interesting.
Yeah.
Great. So I'll invite all our speakers back onto screen. Obviously, thanks, guys, for your time tonight. It's been really interesting. I think we'll open up to questions. A couple in the chat. That one up. So. Yeah, the first one's over to you, Alan. So it's just one based on the samples and diagnostics. I've got one saying, how quick does a sample need to be with your vet/lab when you're sending up for diagnostics?
As soon as you can. I think the basic idea is we would always say to you, like, some of these bacteria can be a little bit problematic to recover. And either way, you want a diagnosis as quick as possible. If it's toxo, you've got a longer haul in front of you. But if it's enzootic, you could potentially look at antibiotics. So even though we've said it's not the common protocol, some people feel they need to do something. If it's bacteria, invariably a lot of the bacteria may indicate risk points, such as listeria, what's the feed situation going on, Campylobacter, it's probably an introduction that you're going to have to ride the storm. But again, it still emphasizes the real importance of how to isolate those sheep and try and reduce the potential for spread and biosecurity. So ASAP, I would always say, once it's been gone down for about 24 hours, the value drops markedly just due to autolysis. And if you look at the weather now, fair enough, it might be sort of like 21, but even in the spring, we can still get some fairly hot days. So rotten lamb sat in the bottom of a bucket will go off pretty quickly. So
sort of post 24 hours, you're not going to get much, or it's still worth submitting? The value drops.
We would still say, look, if you've got placenta, we've seen already, placenta is very useful for looking at things like chlamydophilin toxo, and it's still a very valid sample. but it has to keep making sure you manage your expectations of the client, that if you haven't found a bacterial cause and you've ruled out enzootic and toxo, it might be the quality of the sample's not quite as good. And if it's going to be a problem, and pessimistically more will follow. But again, that just emphasises get them in as soon as you can.
Brilliant, thanks, Alan. I think my next one's going to be directed to Lee. So with the toxo infection, is it spread by rats or are they an intermediate host?
Right, good question there. It's not spread by rats, but it's a good question because rats are often infected with toxo. So if your cat eats the rat, then the cat will, if it's naive, will start shedding toxoplasmosis. So the key thing here is the cat, but it does tend to be younger cats. So I understand a lot of people will obviously have cats on the farm to keep the rodents down. That's a really good thing. And your older cats tend to be absolutely fine. They're probably immune. It's just your young cats are a problem. So yes, rats are infected, but they won't spread it directly to the sheep.
Brilliant. Is there anything, it might not be a question for you, Lee, it might be more for our honorary vet on the call, but is there anything you can do in terms of preventing it in the cats? Obviously with dogs you can treat them with wormers to prevent them passing on parasites to the sheep. Yeah. Is it the same with cats? Is there anything you can do to prevent them getting...
No, good question again, Nicola. As far as I'm aware, there isn't. There have been people working on, a key thing would be to get a vaccine for cats. And there have been some experimental vaccines for cats that were done that were actually very successful, but never commercialized. There's no drugs, as far as I'm aware, that will cure cats. So I think the issue would be, if not the issue, the opportunity would probably be to get a vaccine for cats. I think that would work pretty well, but it would be also then persuading cat owners to use it. So I see Alan's got his hand up. Alan, do you want to add something to that?
Yeah, it's just the fact of One Health. We're looking at, this is a farmer talk, but you consider the number of people that do gardening. There's a lot of people come across cat feces when they're gardening, etc. So the risk to the general public is actually quite significant. Children as well. There's a lot of potential things. Oh, we've got a sandpit for the kid. With a cat poos in it as well. So I think the One Health thing may actually be pushed forward at some point further down the line to say, look, at the end of the day, once we become more, and we are becoming more fixated with potential pathogens that can come from wildlife and even their own domestic pets, people might look to push these things forward. And so vaccines might become more pragmatic approach, shall we say.
No, it's a really good point. And I know when they tried the experimental cat vaccine out in the States, it certainly reduced the levels of infections around pig farms. So you're right, there's a sort of One Health thing. I think if you can tackle the main host, which is the cat, it would make a huge difference, as you say, as well, to public health.
Brilliant. And we might not know the answer to this one, but I am intrigued by it. So do you think they check for toxoplasmosis in humans when they do a post-mortem? Is that like a normal thing that people check for?
That's an interesting question, Nicola. We did some work a while back with, it was called a sudden death brain bank, which is a brain bank source in Edinburgh for people that had sudden deaths, either car accidents, heart attacks, whatever. And interestingly, when we looked, we got small samples of brain from that bank, and we found that actually something like 18% of these people were infected with toxo. So as I said before, a lot of people are infected, but they don't know they're infected. Most people don't show any signs, and the major issue is if a pregnant woman might get infected for the first time, that's a problem, or if you're immunocompromised. That's also a problem. But yeah, in terms of doing, it'd be quite interesting, but we reckon about 1 in 5 as an average. And as you get older, you're more likely to be infected.
Brilliant, thanks, Lee. And then we've got another very topical question coming in about vaccine provision that probably falls with me. And then do we have an issue with vaccine provision? So I know NSA is very keen to make this one of its priorities, and with the new government coming in, it's one of the things that we've put as our list of 8 main priorities, one of which is secure vaccine supply and procurement for the future. To keep people that are on the call in the loop of what's going on currently with the Enzootic abortion vaccines that David alluded to in his presentation. Currently, Enzovax abortion, the next sort of batch was due the back end of this year, and unfortunately there's been a batch failure, so they've not got any new supply coming in until the first quarter in 2025. And the second vaccine, the equivalent made by CEVA, the CVAX chlamydia vaccine, They were expecting their next batch provision in August. It's been delayed until October, but they've no certainty that it'll come in. So there's still the Hipra vaccine available. So there's still 1 out of the 3 available. I know that's not ideal at all because we're coming right up to topping season. But Beth has shared some really interesting some advice from the Sheep Vet Society in the chat about what you should do if you can't get hold of the vaccine. So it's not, there are still some available in the shops, but once it's essentially, we're not sure when the next batch will be coming in. So have a chat to your vet, have a look at the advice that we've shared in the chat as well as come from the Sheep Vet Society about which animals to vaccinate, which are the priority animals, and also when you speak to your vet. So I think I'm going to sort of wrap this up because we're coming up to that past the hour mark and people start to drift off. So firstly, thank you to our speakers tonight. It's been really interesting, really fascinating from my point of view. Hopefully those that have joined today have benefited also. The webinar is recorded, so you can watch back again on NSA's website. We'll put it up with the next sort of 24, 48 hours. But I think my key sort of messages to those that have been listening or are still listening is look at what you're bringing in, and so if you're buying in stock, look at your biosecurity, what you're bringing in, and what's in your flock already, what they're exposed to in terms of wildlife, feed, etc. You're giving them. Check out your biosecurity, get that a top priority, investigate your losses. So make sure you're investigating any losses that come through it lambs or ewes, and just find out what's going on your farm and get your vet involved. They're a great resource. They have a lot of knowledge and experience. And as Alan and Dave were saying, they do all the diagnostic liaisons between those diagnostics and yourself. So please get them involved, whether it's for dealing with something, ideally get your flock health plans in place first with them, and go from there really. So yeah, thanks everyone for joining tonight. Hope it's been interesting, and we'll see you at the next one. Thanks again all.
Okay.
Cheers.
Thank you very much, everyone.
Bye.